top cell, then eventually a long red and a short green chromosome are going to end up in this cell, and a long green and a short red chromosome are going to end up in that one. That's a source of genetic variation. We call it the independent assortment of chromosomes happening during metaphase 1 of meiosis 1 that leads to genetic variability. So we've had kind of two sources of genetic variation here already in meiosis. First, crossing over, or genetic recombination. Second, the independent alignment or assortment of chromosomes at the metaphase plate. Now we're ready to move on, and we'll move to Anaphase 1 and Telophase 1. So let's get those drawn out. I'm going to, unfortunately, have to erase things so that we can draw out Anaphase 1 and and Telophase 1. Once we get this erased, we will draw out the rest of meiosis 1, the reduction phase. Remember, by the reduction phase, we mean that during meiosis 1, the chromosome number is going to be cut in half by the end of meiosis 1. So here we are with, we're in anaphase, right? I'm somehow missing caps to my to my markers. This happens all the time. I lose things all the time. Okay, we're good. We're good. All right, so Anaphase 1 of meiosis 1 goes something like this. Let's draw the cell long and skinny because a couple things are going to happen here. First of all, due to the action of the non-kinetochore fibers in the spindle ratcheting by each other, kind of stretching out the cell elongates. So the cell elongates due to the action of those non-kinetochore fibers that are pulling the cell apart, all right? And the second thing is, what happened in Anaphase of Mitosis? Sister chromatids separated. In Anaphase 1 of meiosis, homologous chromosomes are going to separate and move toward opposite sides of the cell. So the homologous chromosomes separate from each other and move to opposite sides of the cell. Okay, so let's draw that in a minute. If I remember, I had, I hope I remember what I had. I think I had a long green chromosome, okay, on top here, and the short green chromosome on bottom over here. I'm just drawing my colors at the same time. And then my long red chromosome and my short red chromosome up here. So I need to draw in the the spindle fibers so that you can see that these homologous chromosomes are being separated and moved toward opposite sides of the cell. This is kind of cool because now these homologous chromosomes are like, bye, I'll never see you again. This is when we're cutting the chromosome number in half. Well, telophase 1 is when that split actually happens. In telophase 1, all sorts of things can happen. Telophase 1 for different species is really variable. I'm just going to show you an example of what can happen in telophase 1. Telophase 1 is kind of like the opposite of prophase 1. And so all sorts of things are going to happen. But in telophase 1, the spindle may disappear. Okay, so let's write that in, that the spindle disappears, falls apart, all right? Let's write in that the nuclear envelope reforms around the chromosomes. The chromosomes start to uncoil. The chromosomes uncoil. That means they become that stringy mass called chromatin again. All right? And cytokinesis can happen. That's the separation of the cytoplasm here by a cleavage groove, if you will, right? So what we are left with, if these chromosomes move into and are surrounded by the nuclear envelope like they should be, we're left with, in this particular cell, one long green chromosome that is still replicated and one short red chromosome. Okay? And in this cell down here, we have one long red chromosome and one short green chromosome. Right? How many chromosomes are in each of these forming cells? 2 or 4? 2. One green chromosome that's long, one short chromosome. One long chromosome, one short chromosome. 2 chromosomes in this cell, 2 chromosomes in this cell. We started with a cell with 4 chromosomes. We've already cut the chromosome number in half. We've made this cell haploid. It now only has 1 set of chromosomes instead of 2 sets. So we say it's been reduced. There's still some issues here. We still have replicated DNA. We still have, each chromosome is made up of 2 sister chromatids. They're going to have to separate in meiosis 2. And we still have some other things that I didn't draw in very well. Like, remember all that crossing over that happened, the genetic recombination? I didn't draw that in. You might have to draw that in your own notes. Put in, little pieces of red chromosome on that green chromatid. Put a little piece of green on this red chromosome, okay? Because there was genetic swapping that happened back in prophase 1 of meiosis. But at the end of telophase 1, we should have 2 cells. Each cell has half the genetic material, but it's still, each chromosome is still replicated. So we still have to take each one of these cells and go through meiosis 2 to get rid of the replicated DNA, if that makes sense. We'll draw out meiosis 2 in the next video.